for people who are still depressed after Red Sox World Series loss, not yet, and this thing will lift your spirit. And we figured we need something new. So this is called Joint Translational Neuroscience Seminar Series. It's almost like a World Series. So so this is the first time I had it in the morning, so it's great. And this is the kickoff, so I'm very, very happy that we have Gabrieli and to kick off this new series. This is supported by Poitras Center and the Stanley Center, and jointly with MGH. And Pat and Jim Poitras are here, and thank you for coming. And today, our first speaker for this series is Gabrieli. You probably all know very well. He's the investigator at the McGovern Institute, also the Grover Hillman Professor of Health Science and Technology, also the Professor of BCS in our department. And he's also the Director of Martino Imaging Center at MIT. He's also the Director of the Integrated Learning Initiative at MIT. I don't know how he does all these things. I only do once in my lab. I'm still running like a crazy dog. And John, before joining MIT in 2005, he was at a professor at Stanford for 14 years, and so he had won many awards, and he was also elected to American Academy of Art Science in 2016 this year. John's research is focused on actually human cognitive aspect of the real human brain. Unlike many of us, we take easy, we're going to study animals. He actually really study humans, and he mostly uses brain imaging method. You will probably hear him today. He's a pioneer and a world leader in use imaging approaches to study human memory and the language, and really in human object. And his research examines the function and the dysfunction of the human memories in normal object, in normal human brain functions, and also in disease, including Alzheimer's disease, Parkinson's disease, and dyslexia and ADHD. So you probably will hear some of them just today. And he also studies. He also studied the changes of developmental changes, how this the structure and function change during development affect the mental ability in children. And most recently, he has been pioneering something which we think is really exciting. We all know for most of brain disorders, and especially mental illness, we don't have biomarkers, and clinical trials often fail, not necessarily because of bad drug target. A lot of times, we just don't have a way to measure them. For depression, you ask, how did you feel in the last 3 weeks? Right? So it's a very hard science for clinical trial without biomarkers. And John has been doing some really exciting work to use imaging approaches non-invasively, can look at how these mental state change, how the drug can affect, whether can we use these imaging approaches to predict the efficacy or target engagement of drugs. If this works, it will really change the way how we use the biomarkers, not only for diagnosis in the future, for efficacy prediction of even clinical trials. So without further ado, John.
Thank you, Guping, for that very kind introduction. It's an honor to participate in this series and in the research behind the series. And I'm going to talk today about the predictive neuroimaging and precision medicine for neuropsychiatric treatments. I'll tell you what that means a little bit. But my entire career, I've been fascinated as a scientist and concerned as a person who worries about disorders, about the fragile power of the human brain. So we know the brain empowers every human capacity of thought, of feeling, of action in the world. But for every strength it has, it empowers in humans, it has an Achilles heel or a vulnerability. If we're able to think, we're able to have thought disorders such as in schizophrenia. If we're able to have feelings, we can have affective disorders like depression or anxiety. If we're able to be brilliant and invent print, by which spoken language becomes visible over time and space. I had my broken glasses incident from childcare yesterday, sir. From time and space, then we also have brains of individual children who struggle to learn how to read in dyslexia. So this balance between an awesome power that we're trying to decode on the one hand and remarkable vulnerability on the other is the essence of understanding the human brain. And for many years, our knowledge was fueled by injuries, brain lesions to interpret brain differences, but that many things are hard to address that way. But now we have additional tools with lots of limitations, but lots of power as well, with functional and structural neuroimaging, non-invasive imaging of the human mind and brain. So today I'm going to tell you about four groups of studies where we want to ask this question: where might we translate the knowledge that we're gaining about the human brain and its disorders into something that's actionable and helpful for patients and families? What is that bridge going to look like? And so one bridge I'm going to suggest is something about early identification, not waiting until children or adolescents are in crisis and are the victims of their difficulties and things are accumulative, but rather early identifying somebody at risk and preventing or diminishing the severity of the disorder at all. And I'll show you a little bit of work we have that's more promising than the actual treatment. And then second, and Gabrieli touched on this, can we use brain imaging to predict things about individuals that are not currently predictable by clinical and behavioral measures? Can we predict the fate over the next several years of a child struggling to learn to read? Can we predict among patients who responds, for example, to a form of treatment, in this case, cognitive behavioral therapy? And the more we can identify for an individual patient whether a particular treatment will be helpful, we think the more we can steer people to helpful treatments right from the start. Now, I think that the kind of basic research that happens at the McGovern Institute and Broad and elsewhere, that's a kind of research in animals that will, and its translation to humans, that will be the home run game changers, right? Entirely new approaches to treatment. We know that. We need much better treatments than we have now. But you can win a lot of baseball games. And Red Sox could have used a few more singles and doubles, right? You can make people's lives a lot better, I think, with knowing which treatment is optimal for which person now among available treatments. And I will come and tell you in a few moments that how shockingly random it is now which patient gets which treatment. So when I think of infants born into this world around the planet, I think of them as all participants in a neurodevelopmental lottery. They will be born with genes and born into environments that will be more or less supportive for their flourishing and development. And we know that many children face difficulties. About 10% of children struggle to read, dyslexia. About 10% struggle to pay attention in the way that we demand they pay attention. And this is always a fit between what our society or educational system demands and the brain and mind of a child. So before we invented print, there was no such thing as dyslexia, right? Because it was in the brains, but it didn't matter for a person's life. Before we ask that people sit in rooms like this, starting in pre-kindergarten and going all the way through to emeritus status, as I'm working on now, silently in big groups, pretending to pay attention, at least, we didn't demand the kind of focused, sustained attention that we now demand in education for a child to move forward and succeed in the classroom. So ADHD is also partly a consequence of the educational and societal system we live in. We also know that poverty has a big issue in terms of brain and development. We also know that there's risks for many mental health difficulties. Let me switch now for a moment to reading. So my colleagues who study language development, whether it be in linguistics or psycholinguistics, their number one mission is to say, how is it that the child or the infant miraculously acquires language? It shouldn't happen. The parent doesn't have a handbook, the infant doesn't have a handbook, and yet in languages throughout the world, in countless situations, infants brilliantly acquire the language of their caregiver. That kind of spontaneous predisposition to brilliance in language is kind of the opposite of learning to read. Learning to read is the first boot camp that every child encounters in pre-kindergarten and first and second grade, as students, teachers, and parents worry about whether the child gets basic reading skills, which are really the avenue by which all education happens after that. So people in education have the slogan, first you learn to read, and then you read to learn, okay? And without that reading, almost everything in education becomes difficult. And yet, something like about, the numbers vary, about 10% of children really struggle to read. Now, we know something about the psychological causes of this, and like any complex difficulty, there are many different causes that play out differently in different children. But the most common and best understood cause of reading difficulty or dyslexia works like this. When a child learns to read in an alphabetic language like English, they know objects in the world. They know like a book and a scarf. And they hear their parents say the words book and scarf, and they go to school. And then kind of like shocking news about Santa Claus, the Easter Bunny, and the Tooth Fairy. I think everybody in this room is beyond that. I worry sometimes when I talk to audiences with parents and children. Right? Kind of like that shocking. The teacher says, no, you thought that book or scarf was kind of like one big globby word, Actually, there's a secret we haven't told you. It's actually composed of individual sound elements. Those individual sound elements are what's called phonemes. So book has b, and k, okay? And you need to know that because those are the sound elements that will map onto the strokes of black and white on the printed page. You need to know that this sound goes with this sound goes with this letter in order to decode written words on a printed page. You need to know the sound units. And that awareness that words are composed of sounds of unit that can be mapped onto things or taken apart or pulled together has been called a phonological awareness. And so learning to read is largely mapping these phonemic units of sound in spoken language onto the units of print on the page. And up until the early 1980s, people had thought the primary problem in dyslexia was visual. And they thought that because the most common cases were these. The child is speaking satisfactorily at home with the caretaker, goes to school, and all of a sudden struggles to read. And everybody assumed it was the experience with print, the visual experience that was a big roadblock, the big problem, because the spoken language seemed fine, comprehension seemed fine. But studies in the 1980s going forward showed that the most common difficulty for children who struggle to read is actually a weakness in this ability to pull apart the sound units of spoken language into their units. That it's a problem in high-level processing. It's not basic hearing, high-level processing of sound units. There are other things that influence reading development, but this is the best understood. This is our delightful mock scanner used downstairs to make children comfortable and practice going MRI. This is a note from a participant, a particularly enthusiastic one, where she came in. This is her drawing of a noisy scanner, and she writes to the graduate student, "You have let me have more fun in 3 days than I could have in any other place. Just think of it, I'm playing a game when at the same time I'm a research guinea pig. If you're a harsh grader, you take up a couple points for spelling, but maybe you don't." And who knows that when at the same time I'm someone else my age, and if they have brain problems while still earning money, a tiny bit of money goes, to the parent. But here's the beautiful sentiment. What do you think beats that? Nothing, right? Okay, that's practically the motto for those of us who work in this field, which is like, just give us a little bit of a grant on a salary, and then we want to help people, and nothing beats that, right? Okay, but this is expressed completely beautifully by this child. So in a study some years ago now at Stanford with Elise Temple, Russ Poldrack, and others, we looked at 10-year-olds who were either typically reading or who had trouble reading and dyslexia. They came in and we did functional MRI, looked at brain function, and we compared their brains as it's activated by two different conditions. In one condition, they would see pairs of letters like these. They decided if they're the same or different. You don't even have to know what they represent to say if they're visually similar or different. That's our control condition. The comparison condition we're interested in is we show them pairs of letters like these, and for each pair, they have to think about the sound associated with that letter name. So they have to say, do they rhyme? So G and K and in different sounds, those don't rhyme. But T and D and in that E sound, they rhyme, okay? So they're making these judgments about sound. And what we found, and what's been replicated many times since, this is functional MRI, top, bottom, left, and right through 3 consecutive slices up the brain. And typically developing children in activation sort of near Broca's area, sort of near Werding's area as you move up, as these children think about the sounds associated with the letters compared to merely looking at the letters. But look at the absence of activation back here in children with dyslexia and this misplaced one in the frontal region. Very striking differences in how the brains are processing this most basic information. And these are 10-year-olds. They've done a fair bit of reading, okay? But it's such a brain difference in terms of how the print is related to sound. And this core finding has been replicated many times. These are a few of many, many studies that have found reduced response in this area of the brain, in the left hemisphere, around the parietal and temporal area, as children or adults try to think about what sounds are associated with print. We also did a study, and in many ways it was a limited study, but better subsequent studies have replicated this. Same result from the side, the activation and thinking about the sounds of letters from a typical child. The paucity of activation, the reduced activation in the children with dyslexia. And after they undergo an intervention, the growth of that activation here. So when we originally did this, we thought, wow, this is exactly what we think educational support ought to be about. Sort of somehow making available a neural pathway that's typically available, that's not available to some children for some reason, but that the right educational intervention can help them use that pathway that was not available. Now, this is the left hemisphere that I focused on because that's the hemisphere associated with reading and language primarily. But we also had big changes in the right hemisphere. And we have to say something. So we called this normalization because this activation looked more like typical readers. And then there's this huge growth of activation in the right hemisphere, which I'll come back to. And we called it compensation just to have a label for it. That doesn't explain anything. Okay? It's just this makes it look more like typical readers. And this is an unexpected boost of activation in the right hemisphere. Now, we've also been investigating brain differences in children who struggle to read using diffusion tensor imaging, or DTI. It visualizes white matter connectivity in the brain indirectly by measuring micron-level movement of water in the brain. So here's a cartoon neuron. Here's the axon that's transmitting information out of the neuron. Here's the myelin sheath that surrounds that axon if it goes any distance to make the signals go rapidly and accurately. If you're a drop of water floating around in the brain, you can go anywhere you want if you're something like cerebral spinal fluid. But if you're approaching these myelinated axons, which travel in bundles as tracts, you can't go through it, you have to go around it. And that's the sort of indirect way that we estimate properties of white matter connectivity in the brain. And so you can derive pictures like these. Let's see if I get this. Sorry about this. This is working this morning. Not going to work now, but still. It would just spin around. And basically it's showing you that the fiber tracks that are running up and down in blue, back and forward in green, and left and right in red. And so some years ago, around 2000, Torkel Klingberg and I looked in adults, and we found white matter differences in adults with a history of reading difficulty in about the same region that we saw the functional MRI differences. The methods at that time were very limited, but as we could estimate it, it was a pathway that ran forward and backward in the left hemisphere around here, with the biggest difference here. And if we looked at the individual data, and this is a very small number of participants, we wouldn't do such a small number now. This is the measure of white matter property. This is how the reading score in a standardized reading test. The black circles are the poor readers. The open circles are the good readers, the typically developing readers. So we were struck by that we couldn't tell, though, whether this is a consequence or a cause of the reading difficulty. So one interpretation is that this will be the right one, I think, that there's a difference in brain architecture for some reason, that we don't, some genetic reason probably, maybe mixed in with environmental factors, and that makes somebody be a better reader or a worse reader. All right, so that's one possibility. And so this person who had the highest score, they were lucky with the genes and lucky with the environment that made them awesome readers. So that's the causal story. And these children were unlucky as they grew into adults. The consequence story is exactly the opposite, which is this is the brain as muscle metaphor. Maybe everything's about even in all these individuals, but maybe this person was the kind of person who on the bus home read War and Peace in second grade, okay? They're reading. They love it, you can't stop them. And so just like any kind of skill or practice changes the brain through experience, maybe all that reading altered the white matter architecture of the brain. And maybe these individuals who struggle to read do a lot less reading, and in fact they do. And so they just don't have that change. So is it the cause or the consequence? And merely about 15 years later, we have a hint that it's the cause. And this is a study in collaboration with Elizabeth Norton here and Nadine Gabb at Children's Hospital, where we screened approximately 1,500 kindergartners and about 20 diverse schools in the Boston area, brought in about 180 for more detailed testing. And then we're following these children. Now, the critical thing is we're doing the imaging at the beginning of kindergarten before they get formal reading instruction. In Massachusetts, it starts around Thanksgiving. So of course, they had experience with text at home. But in terms of school, they have not yet received formal education about how to read. So we know that there's not a lot of reading experience, and there's no reading instruction. And then we looked in these children, and we focused on the arcuate fasciculus. That's the white matter bundle that connects Wernicke's area and Broca's area, the sort of core language areas of the brain. And we looked at a few other areas as well, and I'll tell you in a moment that what we found is this. If we looked at tests of phonological awareness they have to be spoken tests. These children mostly don't know how to read. So the test of it might be something like this. They might say, listen to the word clip. If I take out the k, what's left, okay? And with any luck, you say lip. That would be an example of being able to manipulate the sound languages, right? The sounds of language. And what we found is, as we looked at the different properties of white matter this way, we found that it correlated quite strongly with their ability to make the sound judgments about language. And this is before any reading instruction. And my colleague Nadine Gabb has now shown that for infants born to a family with a history of reading difficulty, she can see in this same track differences within a month of birth. So we think this is a beginning point for some of the difficulty that goes with learning to read. And so here's where we're now tracking these children through first and second grade. And here's an example of one child who has a poor phonological awareness, and by the end of first grade is well behind her peers in reading. Here's a child with a different white matter size and architecture who had good phonological awareness at the beginning of kindergarten, and by the end of first grade has a good reading score. So what we're seeing, we think, is the beginnings of what will make learning to read easy or difficult. And what we would like to imagine is that now you could do some sort of intervention in a pre-kindergarten child before he or she ever fails. Not wait until they're 1 or 2 or 3 grades behind in reading level, but do a different kind of educational support for them even before they fall behind their peers. So we both want to understand neurobiologically what's different in a child who struggles to read, but also we think it has implications for educational policy and practices as to how to help children succeed and not fall behind their peers. Let me turn to depression for a moment in childhood. And as much as there's been fantastic progress in many aspects of research regarding the brain basis of depression, the reality is that there's still a huge struggle to help children in the rate that we would want to. So here are statistics about the increase in suicide, which is obviously the most tragic failure of mental health in adolescents. Threefold increase in girls of 10 to 14 over the last 30 years. So there's one marker of how important and difficult the challenges of supporting mental health in adolescents. This is a statistic of that kind. Now, there's been many studies looking at differences between adults and also some with children who do or do not have depression. There's many studies like that. But again, we have the same problem as the cause or the consequence. If we're aiming to move towards early identification and prevention, when we see brain differences for depression in a 20-year-old, a 30-year-old, a 40-year-old, are those brain differences the reason they got to be depressed or are vulnerable to depression? Or are they the manifestation of depression itself? And we can't pull those things apart. So one research strategy to understand whether brain differences are about the cause or the consequence of depression is to look at children born to a parent with depression. A child born to a parent with depression has a 3 to 5-fold increased likelihood of developing depression. That means many won't, but there's an increased probability they will. And so, and in our study that I'm about to describe to you, all of the children had at least one parent with diagnosed depression, but none of the children were depressed themselves. But they're all 8 to 14, and about 15 and up is the age when depression most often, not always, but most often begins to express itself in larger numbers. So we're looking at an age range where depression is sometimes but rarely manifested, but it's coming up in these children, the high-risk period. And so in this study, we looked with Jenny Chai and Joe Biederman, we looked at children who were at risk but had no current depression and, with one parent. And then we had children who were the comparison group who were matched in most ways, but they had no parental history of depression. So nobody's depressed clinically, but some children are at family risk and some are not. And while they're in the scanner, what we're showing them is pictures like these where they're matching up whether these faces the same or not that are negative fearful expressions or happy expressions, neutral expressions, or objects. And I'll just summarize the results of the study this way. For the children who were at risk, who had a parent with depression and are at increased epidemiological risk, their brains had a decreased response to happy face and an increased response to negative faces. Okay? So if you think about the cognitive bias ideas of depression, that part of what leads to depression is a focus on the negative and a sort of disinterest or non-response to the positive. That's exactly what we saw. The children at risk, although they were not themselves depressed, their brains were more responsive to negative facial expressions and less responsive to positive facial expressions. Exactly what you don't want in somebody, a focus on the negative and a sort of disinterest or lack of response to the positive. We also looked at two things. I'll just mention them very briefly. Diffusion tensor imaging, which I told you about, to look at white matter connectivity, and then resting state functional connectivity. There it's a functional MRI measure. We're looking at naturally or spontaneously occurring fluctuations in brain responses or the fMRI signal while the child is there doing nothing. Okay, there's no task. We're just looking at spontaneous relations between regions. And without going into detail, I will just say that we found multiple differences, both more connectivity and less connectivity, including the subgenual anterior cingulate cortex, which is sort of the hotspot for depression research in these two groups. And the thing I want to leave you with is two last thoughts on this topic. One of the things we're really interested in, and I'll move to this in a minute, is when can brain measures tell us things above and beyond clinical information that's now available? And so one of the questions we asked is this. If we use a clinical rating scale that's widely used, the Child Behavioral Checklist is widely used in pediatric psychiatry, how well can we differentiate the children who are born at risk versus not? And if we look at our functional imaging differences, how well can those tell us apart? Okay, so because in the end, we're not going to want to look at children who are just born to families with a history. We want to look at all children and discover who's at risk and where we might intervene early to avoid stark depression. And what we found was that if we look at the CBCL rating, a clinical checklist that's the kind that's widely used in research, this was our accuracy, 64%. But with the brain, we were able to better differentiate the two groups. Now we have a lot to go on that because we don't know who will develop depression and who will not. But this is one hint where the brain measures seem more sensitive to risk than a typical checklist. And the last thing I'll tell you about this is just very briefly is we also looked at white matter development across the ages in these same children and found something striking. Now it's a cross-sectional study. We're looking at each child at a certain age, and we need to do longitudinal ones, but cross-sectionally. We found that we look at some of the white matter around the anterior cingulate, we see an increase in this property of white matter that typically increases in development in the control children, and a starkly different reduction of the signal in the children who are at risk as they approach the very age where they're at highest risk for starting to develop depression. So we see a very different developmental trajectory in these children in the organization of this white matter as well. So there's a lot of different ways in which I think we're going to get signals about who's more at risk and understanding maybe with whom we ought to intervene and maybe even having an immediate measure of whether the intervention is working or not. So now let me turn a little bit to treatment. So most neuroimaging studies, including the ones I showed you today, are basically sort of correlations of one kind or another between differences in brain signals between groups, like patients with or without depression, people with or without depression, conditions like showing faces versus words, or something about behavioral performance in the scanner. That's what most of us do all the time. But I'm really interested in terms of thinking about clinical challenges and helping people. Are we at a moment where brain measures tell us something powerfully more than current behavioral and clinical measures? And this is always, you have to do this with one hand ready to erase, because tomorrow somebody can have a better clinical measure or behavioral measure. It's not like the field is going to stop, but it's just where we are now. And so what I want to tell you in the last second half of the talk is basically, can we use prediction, and I'll use it a couple of different ways, as a humanitarian and pragmatic contribution from human cognitive neuroscience? So when you talk about prediction, on the internet they have funny debates about who said this. Prediction is very difficult, especially about the future. Depending on which source you see, it's either Niels Bohr or Robert Peterson or Mark Twain. Or I like Soren Kierkegaard's thing, life can only be understood backwards, but it must be lived forwards, all right? So in a certain way, that also the results I'm going to show you are backwards because we know the outcomes. And then we're going to look back at the brain, all right? And in the long run, we need to run the other way. We need to look at a child or a person and say, oh, you're at risk, we should help you. Or, oh, you would respond better to this treatment than that treatment, right? But at the moment, we're still doing this. So I'm going to show one example from dyslexia and one from social anxiety disorder. Now, I'm a huge fan and participant in the community of human cognitive neuroscience. So we learned a ton about functional and structural architecture of the human brain, how it varies across people and cultures, how it varies in neurodevelopment and neuropsychiatric disorders. But we have one huge challenge, I think, in our shoulder, which is, to my knowledge, and I'll make this case in a moment, and then maybe I'll get beaten back, we've made nobody's life better. No patient or family or clinician who wants to help a person is better off right now based on everything we know from functional or structural human neuroimaging. It's kind of a shocking thing. I'll come back to this and try to defend it, and then so I'm interested in what kinds of brain measures might be different than that. Can they, for example, better predict long-term outcomes? Can they better predict individuality, variance from patient to patient by who responds to a treatment? So here's an amazing thing. If you go into PubMed and you put in a diagnosis like schizophrenia and MRI, you come back with almost 6,000 published papers in PubMed, right? Or depression or anxiety or autism or ADHD or dyslexia or obsessive compulsive disorder. You add those up, you're about 20,000, and that's not including EEG, ERPs, positron emission tomography, diffusion tensor imaging necessarily. Okay, so 20,000 publications, and yet, when DSM-5 came out in 2013, not one diagnosis, not one discussion of treatment has been influenced by anything we've done in brain imaging so far. And it only makes me feel better. I'm a huge fan, of course, of the amazing genetics, all over the world, but it broad outstandingly. I don't think genetics has done any better yet. Is that, okay. No, it's like you're the drowning person. You want to pull somebody with you. Let's pull down the genetics. But seriously, really, we're surrounded by the world's outstanding hospitals here, and nobody is diagnosing or treating, I believe, any brain difference or learning difference fundamentally different based on anything we know about brain or genetics. So I think that's, and so the challenge, I think, in part is to say, what could be a finding that in the foreseeable future could touch the lives of people, short of some amazing meteor discovery that changes the world, okay? And that could happen. But what could be, and so I'll tell you about the direction that I'm thinking, and other people are as well. And let me just say one methody thing for a moment. Usually, and I showed you some correlations earlier, people will correlate things like, for example, a brain signal versus the outcome of a clinical treatment. Most of those are what people kindly call overly optimistic. That is, any given correlation you get is inherently unlikely to be replicable of the same strength for a new group of participants. It's just the way things work typically. So the core analyses I'll show you now aim to be one step better than that, which is using methods that on purpose seek generalization and individuation. That is, for example, leave one out, cross-validation, in which each person's outcome is predicted on the basis of the activations or the brain signals from the other participants in the study. Because if we're ever to apply these things, to help patients, it has to be for a single patient, and it has to be in the moment that they're choosing what course of treatment to get, right? So we're very interested in these questions about how to make these kinds of predictions better. And I'll say two things about these neuroprediction measures that are different than we normally do in studies with patients with mental health differences. First, most studies look at similarities among patients. If you compare a group of patients with schizophrenia or bipolar disorder or depression to typically developing individuals, for example, you're interested in what's the most similar thing about each group that you can pull them apart. In the studies I'm about to show you, we're interested in the opposite. We're interested in heterogeneity among patients, because it's a heterogeneity we want to understand as it comes to, for example, to their clinical course or response to treatment. So it's exactly the opposite. We want to understand diversity, not similarity within a diagnosis. And the second is we're not really studying anymore brain differences that go with a disorder. I think we're really studying the brain's capacity to heal. That is, if we're studying response to treatment or progress in an area, we're not necessarily studying the reason why a person has a difficulty. We're studying the brain mechanisms available to them to get better. And those things will be somewhat related, but they're often not identical at all. Right? So I think that's a kind of almost a newish field in brain imaging, which is to say, let's identify the neural mechanisms in the brain that are available to help a person get better. So let me start with dyslexia, and then I'll talk about social anxiety. So we know some children with dyslexia make considerable progress, and some don't. What's the brain basis of that? Can we tell who's going to progress and who won't? So in the study with Fumiko Heft, we have somewhat older children, around average age 13, see pairs of words like bait and gate and ask if they rhyme. To make it a little tough for them, we make them look visually different, so they have to sound out the word again. And so that's how we're looking at phonological processing in the brain. In 25 children with dyslexia, 20 typically reading children, we do the brain imaging, functional MRI, diffusion tensor imaging, and we give them 17 behavioral measures, IQ, reading, and language, that are widely used in clinical and educational research. And then we see them 2.5 years later in a longitudinal design and see how they're doing in terms of reading. And here's what we find. So here's a single word reading score. 100 is a population average. Here's our controls. Now, you could say, what kind of public school system is not moving these children to be any better readers in 2.5 years? But it's okay. It's not a critique of education because the scores are age adjusted, okay? So these children are reading well for their age, and they remain reading well for their age. Here are the children with dyslexia reading poorly, but one group makes some progress, and one group makes no progress relative to their age. That's single word reading, which was our identified outcome measure. But if we look at comprehension for our paragraphs, which is the ultimate goal of reading, not getting a word correct at a time, but grasping the essence of a paragraph, you can see these same children who make some progress on single word identification make a lot of progress on grasping what's in a paragraph. And these children make very little. So we have two subtypes, if you will, of children with dyslexia, those who make substantial progress and those who don't. Now they're doing this task, deciding whether words rhyme or don't. And we have a big surprise for us, which is that the part of the brain that was the best predictor of which child would be in the group that makes major progress versus not is activation in the right frontal cortex. Each dot here is an individual child with poor reading in red. The more they improved on reading over the years, the more was the response in right prefrontal cortex as they judged whether pairs of words rhyme or didn't rhyme. I'll come back to that in a moment. I'll just say here, without going into detail, that we also found similar structural differences in right prefrontal cortex using diffusion tensor imaging. So that makes a bit more confidence that it was both functional and structural right prefrontal cortex. Now, why is that weird? It's weird because one thing we know about learning to read in typical development is as children begin to read, they're pretty bilateral. They use both hemispheres to begin to deal with print. Because as you're a beginning reader, print is a visual spatial puzzle. Is it a B or a D? If you've had a little kid recently you've been reading with, it's like the hardest question you can get at a presidential debate. Is it a B or a D? Is it a B or a D? And they go like, there's no way to know, B, okay, and the T's and F's and all that kinds of stuff. They're very difficult visuospatial problems, and the right hemisphere is dominant for visuospatial processing, and it's very bilateral. But as children get better at reading, a few grades later, a few years later, almost all the processing of letters and print becomes left hemisphere dependent, because that's the way it enters the left hemisphere language system, and a fluent reader directly relates the print on the page to the language they know. Okay? So that's leftward. Another thing is several studies have shown that as children become more fluent readers, early on they activate both frontal and posterior areas, but as they become better readers, it tends to move posteriorly. And that's because that frees up the frontal cortex to do things like thinking, inference, and so on, because now you have a rapid automated system for mapping what you see onto onto sort of language systems. So we see most of simple processing of print moving leftward and back, and yet for those who improve the most, it moved rightward and forward. And furthermore, if we did a multi-voxel pattern analysis, a form of machine learning to try to distinguish which group a child belonged in, the ones who improved or not, we got the sort of random set of voxels we usually get, which we don't know what they mean. That's just looking at all the voxels in some huge machine learning data-driven way, including some in right prefrontal cortex, but in other places. So I'll summarize the study here. Here's a key thing. None of the 17 behavioral measures predicted whether a child would improve or not, either categorically or continuously, none. And at first we thought, well, we're brain images, we don't know the education literature if we went out there, but I can tell you, when talking to many clinicians and looking at every journal we can look at, nobody can predict which 10-year-old will make big progress and which will not. And our best predictors here, we're at 90% accuracy about in knowing whether a child is in the group that will improve or won't improve. So there's two messages here that I think that we think about from this, or three. One of them is, when a child gets a certain level of reading, are they better off, on average, trying to read like other children, left hemisphere pathway, or trying to read in a way that's best for them, given their brain architecture and whatever path got them there, right prefrontal cortex, right? A suggestion that the activation I showed you earlier, all this right activation, right hemisphere activation response to remediation, might matter a lot and might matter even more. So we always thought the point was to make poor readers read like good readers, but maybe the point is to let poor readers read the way that is best for them using an entirely different brain pathway. And the children who don't succeed at that are the ones who do the worst. And the second, if you told a mother, gee, don't worry, and we're far from this, but not, hopefully not more than a decade, don't worry, your child has a 90% chance, I'll make up a number here a little bit, that he or she will make great progress in the next few years in reading. For every mother like that or father like that, you would tell another parent, oh, I'm sorry, your child is in the group that won't make progress. But here's the important thing. Those children should get a different kind of intervention. We shouldn't sit back and wait, as we do, for half the children to fail and then go, oh, gee, glad we helped some, couldn't help others. Those children whose brains are different enough and in ways that we can measure, we should be very aggressive in trying experimental interventions that don't just wait for failure, but try a different approach. For the half that benefit from education as it now operates, great. For the half that don't, something incredibly different is needed, not just waiting for failure to play out over the years. The last study, or set of studies I'll show you about is social anxiety disorder. So when I talk to my colleagues in psychiatry, who I collaborate with, and I have wonderful ones at multiple institutions in the Boston area, and you ask them, how do you decide which medicine to give? Or how do you decide for a given disorder? Or how do you decide whether a person should also get cognitive behavioral therapy? One of my favorite answers is doctor's choice. Okay? Some people practice genetics in the following way. They say, oh, I ask if anybody in your family has that disorder, and if they liked a certain drug, we'll give you that one. Okay? And that's not bad, maybe. I mean, but it just tells you how physicians, patients, and families are operating in complete darkness as to which treatment is appropriate for which individual. And a hint about how big a problem that is this. This is the famous STAR-D study from NIH looking at depression. And what they found is in 4,000 patients, the first treatment they give, one treatment, about half have a positive response. Then they try a second drug and a third drug and a fourth drug. By the fourth drug, 70% have had a response, okay? But people are dropping out a lot from the whole study, even though the drug is made available to them, because they're dealing with life for not moving forward with a treatment that didn't work. They're discouraged or other problems arise and so on. So here's another study comparing drugs versus, for depression, drugs versus behavioral therapy. And again, about this 50% response rate in terms of effective therapy. And that's almost the number you see all the time. For any well-supported treatment that's gone through FDA approval or things like that, about half of patients have a good clinically useful response, and about half don't. And nobody knows which half you're in when you go into a doctor's office looking for help. So, so in one sense, sometimes we have tough problems, like how does the brain work? That's a tough problem. All we have to do here is significantly better than chance, and we could provide something useful to families and clinicians, right? The bar is low. Now, it's not so low that if we have, it's not so low that if we only get like 1 or 2 percentage points better, and we can publish it, but it's not practical. So we have to move, but it's low because now treatment is randomly assigned. So I'll tell you the final study, social anxiety disorder, an excessive and unreasonable fear of social situations, 7% in a given year, chronic disabling disorder. This is severe social anxiety. And to give you a sense, twice the rate of unemployment versus other anxiety disorders and depression, because so much of getting a job is about interacting with a person. This is not shyness. This is a crippling fear of interacting with other people. Treatments, again, sometimes drugs, sometimes behavioral therapy. About half of patients respond to each. No clue as to which patient is going to be in the half that responds to either one of these poor predictions. So in this study, what we did is we looked at 51 patients before and after cognitive behavioral treatment, and asked whether how our measures would go. And now we have to be competitive. So one measure of the severity of this, a clinical measure, is the Leibowitz Social Anxiety Scale. That correlated 0.38, or accounts for about 16% of the variance. So severity, the more severe your disorder, the more you respond. But we have to beat that, and that's considered so useless that even though researchers will measure this, no clinician will bother to use this kind of information. And I'll just tell you two studies. In the first study, we had individuals in the scanner looking at baseline before they get the treatment, seeing faces that are negative or neutral, and scenes that are negative or neutral. The scenes were irrelevant, the faces were, which makes sense for a social disorder, right, or a difficulty. And without going into great detail, what I can tell you is activations in posterior visual areas correlated quite strongly, far higher than the severity scale, with how much an individual patient benefited clinically from the cognitive behavioral therapy. So there's much information there somehow about whether a patient is likely to respond strongly, moderately, or weakly to this kind of treatment. And although we looked at it mostly continuously, just to simplify the picture, this is activation for negative faces compared to neutral faces in the half of patients who had the most response, and this in the half of patients who had the least response to the cognitive behavioral therapy. So there's all kinds of things we can think about that and need to learn about that, but we're way higher than the clinical rating scale. And the last study I want to tell you about very briefly put us higher than that. So we both looked at diffusion sensor imaging as a measure of white matter connectivity and functional connectivity, fMRI signals. And we put those together. And the appeal of those two things is that they're really easy to give, relatively speaking, across institutions and scanners. All you have to do is lay still long enough for us to measure, which is not trivial for everybody, but there's no task involved, there's no task demands, there's no arguments about which stimuli to use. You're just laying still for two sets of scans. And in this study, we looked at connectivity to the amygdala, a sort of a whole brain multi-voxel pattern analysis, which is data-driven. We looked at the white matter properties of the right inferior longitudinal fasciculus, the white matter tract that's closest to that part of the brain that showed the activation differences in the prior scan I showed you. Here's an example of the brain differences we saw in relation. Here's the improvement, the clinical improvement. Here's the white matter properties at the beginning of the treatment. And you can see that the more they have a certain white matter property, the more they benefited from the treatment. So if we put our measures together, here's how much of the variance we could predict by clinical severity on a formally administered rating scale, about 12% of the variance in the study. Here's how much we do if we add that to diffusion tensor imaging. Now we're moving up to that high. Here's how much if we look at the resting state measures plus the severity, and here's how much if we put it all together. So now we're counting over 60% of the variance, which I think is meaningful. And the other quite striking thing is that all these sources of information, at least in this study, were additive. We thought it could be redundant. You could be measuring the same thing 3 different ways, so you keep adding them and you're not moving forward. As we measured these different properties of the white matter and the resting state and put them together with initial severity, you're now at 60% of the variance in whether a patient responds powerfully or responds minimally to the treatment, the range of responses. Sometimes people want to say, well, for an individual patient, we need green light, red light. Don't tell me something about 40% of the variance. I need to tell, So we said, okay, let's make, this is always trouble when you draw a line, but let's make 50% improvement on the LSAS. That's a sort of standard one to say, like, do you improve enough or not? And with the logistic regression with these brain measures and the initial severity, we're about 81% accurate in knowing whether an individual patient is in the top group in responding to the treatment or the bottom group. So we think we're in a range that is well above chance. And now this is just one study. It needs replication and generalization. It needs stronger models of single patient outcomes. And of course, the decision being faced by a family or a patient or a clinician is not giving a lot of information about one treatment. It's which treatment to pick among alternative plausible treatments. And so we're terribly interested in that, but there's beautiful work coming out from Helen Mayberg and her colleagues for depression Emory. I'll just tell you very briefly, they show the differences in the brain in positron emission tomography scanning that identify patients who responded especially well to CBT versus especially well to a medication, so different courses of treatment. And we think a lot of the future is going to be about that, which is to say not any treatment, of course you're going to help somebody, but which among plausible alternative treatments is the most likely to help somebody. So I've showed you examples of, I've summarized in a review paper published a year and a half ago or 2 years ago almost now, about 70 studies that with weaker data or stronger data, it varies a bit, all predict things. And let me tell you about two of them. So prognosis for depression, response to CBT or medication for depression, response to medication anxiety, response to cognitive behavioral therapy and schizophrenia. Let me tell you the last two. And the bad thing about the last one, I'll tell you in one moment, is that that's the only one most of you will remember after this session. Okay. Here we go. Yeah. So this is a really interesting study from Martin Paulus, which I wish there were many replications of this attempted that I'm not aware of, who looked at patients coming out of a 28-day program for methamphetamine dependence. Now, 28-day program is a fairly standard number for inpatient treatment for substance difficulties. It turns out that that number 28, there's theories around, but no evidence where that comes from. Why not 21? Why not 35? There's a theory around that because the military would only give 28-day leaves for soldiers, but there's no evidence for any of that, okay? So we know that 28 days is probably too long for some people and too short for others, right? And so what Martin Paulus showed is that looking at 28 days, if he did fMRI on individuals as they came out of that treatment and then tracked them for the next year, he could identify 37 of 40 whether they would or would not relapse. We need a lot more science on that. But if we could have much more certainty whether a patient needs 2 weeks, 4 weeks, 6 weeks, 8 weeks on a scientific basis, I think that would be phenomenal improvement in terms of how we approach treatment for substance disorders. This is the one you'll remember, and this needs a lot more work also. So this is a study where they did imaging as parolees came out of prison and asked whether they could predict who would be rearrested for offending in the next year, and got results that were considerably above demographics like age and criminal history. There's some debates about the precise methods, and you certainly wouldn't want to. It's like science fiction, right? They could predict decently who would get rearrested or not in the next year. And of course, that's exactly what you want to know in a parole hearing: is should somebody stay in jail because they're a threat to society, or should somebody be released because they're ready to go back to a life that's productive in society? And you could say, well, we worry a lot about getting that right, and you would. So there's a lot of social, the more we get good at predicting people's futures, the more social and ethical issues there are to think through about how to handle that information responsibly. But let me tell you one last study, which I won't show you data, don't worry, which is a study of judges in Israel deciding parole that was published some years ago. And it turns out a fairly significant factor was the time of day your case was heard. If you were heard in the first half of the morning or the first half of the noon after lunch, first half of the afternoon after lunch, you had a better chance of being released on parole than the second half of the morning or late afternoon. And the hypothesis is, nobody knows why, the hypothesis is that to make this hard decision to allow somebody out who's a criminal offender from jail on parole, you need a lot of mental energy because it's a tough call to let them out. First thing in the morning, those of you know this, you had your coffee, you're ready to go, you're thinking hard, you make the hard decision. Second half of the morning, you're fading is the hypothesis, and you go, put them back in jail. It's too hard to figure out. You have lunch. You have a little bit of mental energy again. So first half of the afternoon, more likely to decide for parole. Second half of the afternoon, people are fading, less likely to decide on parole. So I'm just saying a ton of decisions about people at so many places in so many ways are made on the basis of very arbitrary and poor information now, whether it's what treatment they get, or parole decisions, or things like that. And so if we could be somewhat more scientific in understanding what's the probability, given knowledge about a person's brain, that they would benefit from a particular treatment, for example, I think we could do a lot for people as we wait for dramatic improvements in treatment. What if we could get much more accurate at sending the right patient to the right treatment now? So I have terrific collaborators. And I'm very grateful for the support from the Poitras Center for Affective Disorders Research at the McGovern Institute. Without that, we couldn't have done a ton of this work. It's been incredibly valuable, as well as support from the NIH. Thank you very much.
Open for questions?
Yeah. I'm going to call? Sure. Yeah.
The patient's activated in brain regions, for example, compared to the control. Why is that? Do you think this is basically physiological change, which is, for example, could be genetically determined, or do you think this is some kind of environmental or random? I don't know.
So we like to tell ourselves when we have a good result on these predictions, that the advantage of the brain is that we pick up the advantage. practically, but the disadvantage scientifically is that we can't differentiate at all the genetic piece versus the environmental piece. Okay, so they're both in the brain, obviously. The genetic, predispositions and all the effect they have, the environment one has, all of that's in there, and we can't tell those things apart. So that might be good for prediction because you have estimates out of both the genetic and environmental contributions, right, to a person's brain and mind. Bad for knowing, what the pathway is when we're so far from pathways.
So you never did like a family study where you would look, for example, if there are endophenotypes in the family and some kind of degree of.
No, but that would be, that's a direction a lot of people want to look at. For example, look more at the parents to think about endophenotypes that way. I think this intergenerational research is taking off for the reason you're describing. And personally, I would love it, if we could add. Every time I give this talk, practically, people say, "Well, what about the genetics? Why can't we use that to help predict which person benefits from which treatment?" And I think it'd be cool to try that. I know there's a huge risk in that kind of research, without huge samples. And of course, the issue is we're almost the opposite of huge sample research. We're trying to think about ways in which, for an individual person, we can give a decent probabilistic estimate as to whether they're on a risk for depression or dyslexia or will respond to a treatment better or worse, right? So we're trying, yeah.
But I mean, there are good examples for aphasia where a single variant has a large effect.
I think it would be fantastically exciting to have collaborative things between McGovern and Broad or other institutions and other researchers to see whether we could maximize even more of the variance, looking at the combination of genetics and brain measuring. I would love to do that. Yeah. Well, so I think we've taken like 2 or 3 steps in the right direction, but so here's a weird thing which is how slow science moves in terms of helping people, right? It actually is a brutal thing because even this thing, which is pretty simple, current measures of brain imaging, we're not doing anything new really. Current treatments, nothing new, right? Nobody has to discover amazing things in the laboratory or go against the odds. But to even have enough research, I think, to justify influencing treatment, it's hard to imagine that it would be less than 10 years, which is because you need, a big study. You need to do that, and you need to probably ideally have patients who you build a model out of, and a completely new set of patients to see that it all works. Okay, so it's very, in that sense, it's a little discouraging how much work would need to be done. And that's assuming you got the grant on the first outing at NIH or something like that, and then, even that could be 5 to 10 years. So, but I do think that at the same time, there's so much promising research happening in this way that if we really, give it a big push, it might be sooner. But that's a big path to go. Yeah, Pat.
Is there a specific definition of CBT, an application of the same, or is that a big variant?
It's a pretty big variant too. This is why clinicians often like pills, because they go like, "That's the pill," And when you talk to the people who are very careful about CBT research, the ones we work with, they have what they call manualized CBT. That is, all the people who give it have lots of training to give it the same way to everybody, so it's very controlled. Then as you get out in the community, and this is just real life, there's no rules about who gets to. CBT is so well documented to be effective as the best medication for many things. Obviously, if a patient in the moment of psychosis, you can't do CBT. But if a patient is, in a moment when they can respond to CBT, every study shows it's at least as effective as the leading medication statistically. Right? So it has a great scientific reputation that's well-founded. What you can't control is how every community clinician will say, "I do CBT," because they all want to say that because they're all impressed by that, but in a lot of them, it's just a slogan that's vaguely in their treatment, and you can't control it. It's not like the pills that you hope are manufactured consistently. So this is, so on the one hand, some people will say, well, CBT is doomed in some way because it can't be, On the other hand, if you have a child, a 10-year-old who's at risk for depression, I think some sort of behavioral therapy is the only route that we would feel comfortable in giving it a try at the moment, okay? And so, we just have to try our best. But you're right, CBT, as it diffuses out in the community among clinicians, who it can a lot of it is not what people meant, but Aaron Beck and others mean by carefully controlled and operationalized CBT. So it's a practical problem of delivery. Yeah. I'm sorry, and then after you, I'm sorry, yeah, yeah.
Yeah, part of the reason for practice like that in communities is relationships are a powerful variable. The trouble is it's heterogeneous. I think the thing that I find myself wondering as I listen to the research is why there isn't more attention taken to what goes on in the gap between narrative and biochemistry. I mean, if you think about schizophrenia, there's a good example. There are a lot of things we know about neuropsych function and also therapeutic interventions. So it seems to me as though looking at the relationships between the constructs that have validity might be a way of controlling more of it and examining what actually works, what changes, what doesn't.
Mm-hmm. Yeah, I completely agree with you, and that would be another piece of, in my mind, of this sort of individuality, understanding more of those. Yeah, I think the more we understand that internal dialogue and relationship between a therapist and a patient, we might also make more progress in understanding what that needs to look like depending on the patient. Yeah, I completely agree with you. Yeah.
Yeah, I was wondering about the responders and the non-responders, and the neuropsychiatric perspective. And what are the consequences if you think about non-responders? What are you doing with them? Like, I don't know, if they're not responsive to current therapies or.
Well, so here's what I think. So what the STAR depression study showed is that by the time you get the fourth medication, 70% of patients have a positive response. If you hang in there through 4 treatment failures, right? So if we could know up front, this is my, which medication is likely to work statistically for an individual patient, maybe they don't have to go through 3 treatment failures or 2 treatment failures or even 1 treatment failure, right? So we have lots of examples where we know already that some people respond more to one than another. We just don't know which person it is up front, so they can be initially assigned the treatment that's most likely to work for them. So I think if you're a non-responder to CBT, probably that wasn't a good investment, or it needs to be a different form of CBT. But rather than just send people off the cliff into some treatment, if we knew up front that there's a really good chance that the patient would benefit more from this drug than that drug, more or less from CBT, then you might rationally pick the approach they take. So I think where you have multiple treatments, you could just pick the best one on some statistical, informed, scientific, rational basis. Is that okay? Where you have complete non-responders, that also is interesting, because what happens to those now? When there's a new drug that's tried, right? They'll just be in your sample. They'll be in the non-responders, but if p.05 you're better than your alternative treatment or placebo, the drug is approved. So by identifying something about the brain differences in people who don't respond to any current available one, then those people should be studied separately, right? Otherwise, they'll just always fill in that bottom part of a clinical trial or an educational intervention, right? The children who are not improving on reading, people go, well, what can we do? They didn't improve. But no, but we should try something different with them, because what we now do in schools won't work for them. Okay, so by identifying in some way that people who don't respond to anything, the grimmest group in that sense, in the way you're describing, we also have a chance to say, let's try completely different things with them, and not have them just go, you just have this group that's not responding to one thing after another, hidden statistically in our total group effect, where by the people who do respond. Is that? Yeah, so in some cases it's just an alternative treatment, in some cases it's a new research question, but with the right people. That 20% of people with major depression who don't respond to anything, those should have an entirely different aggressive research approach, because otherwise, why are we sending them to treatments that are highly unlikely to work for them? Is that okay? That's, yeah. Yeah. There's got to be something different. I can tell you, we had the 17 measures of all kinds of reading scores. We could not find any difference. They were equally severe at the outset in general reading problems. So we could not identify anything behaviorally that distinguished those who would improve versus those who would not. But I completely agree with you, that could just be that we don't have the right behavioral analysis, even of the items in the tests we have or new behavioral measures. I mean, there will always be improvements and other perspectives about what to look for. So it's not like we want to say, behavioral analysis, sophisticated one as you're describing, is frozen. Now we compare it to brain things because maybe there'll be other shrewder things, and maybe scientifically go back and forth. Now we have a group of children who won't improve. Now we could explore in them what pattern of errors do they have that differentiates them. So I think each time we discover a group of children who are not responding, or adults, it's a new research question about what's different about them and what then might help them. Yeah.
Yeah, so the group of dyslexic children, they improved in reading and they had an activation in the right hemisphere.
Yeah.
So how do you go on there? Do they have like a interpretation of, do they do it like visually or do they find their own way?
Yes. So sadly, this is the slowness of research. We're working on that, but we don't know that, right? Parents always ask me the question you're asking, like what's going on in that part of the brain and how do we get people to use it? And we don't know. A guess is something like, there's a guess, I have a guess, this is not scientifically supported, that it might be more of a visual approach to reading than a sort of language sound one. And that might be more efficient for those children, because sometimes I talk to children adolescents who've improved quite a bit, and here's what they tell me. If they just get to scan text, they feel like they're sort of doing okay. But if they have to read out loud, they fall apart, okay? What does that mean? That means the typical connection between print and sound is so problematic for them, but they find a better alternative pathway. Maybe a bit like speed reading, actually. That's better for them and it can help them, right? So yeah, so something like that might be right, but we don't know, and we don't have any scientific knowledge on that. Yeah.
Actually wanted to weigh in on that one or discuss, since that's basically the music center, right? That was right Broca's. And the first task you showed was a rhyme task, so certainly that would go along with having the sound out, listen to the sound, make a judgment. I didn't follow there, I missed one thing as to how you got to the paragraphs, what exactly they were doing that was different. But again, if there was some matter of auditorily rehearsing to self, then recruiting right homologue of Broca's area makes a lot of sense.
It's something along that line. And I think the fact that these children who got better got a little bit better at reading single words accurately, but a lot better at getting the gist of a paragraph, tells us that their strategy is not about getting each word correctly. Okay? Their strategy is something about getting the words close enough and paying a lot of attention maybe to context and so on that lets them grasp the gist of the paragraph. And of course, if you get the gist of a paragraph, that's the most important thing typically for reading, for learning.
And the other thing I wanted to say is because that's fMRI and I assume a group average,
yes,
then it's a matter of what averages well to a group, so how focal that activation is and stereotypical, as well as what gets recruited. So there was some work back when I was following this more that we recruit the right homologue of Broca's area for harder language. So it's like we do well enough over here, but when needed, we can recruit this guy over through the corpus callosum. It's a little bit slower to go across, but when needed. So if that was, I can't quite grasp this, but let me sound this out and rehearse it to myself, or whatever it is, the compensatory strategies you were talking about, that might recruit over to the right.
Yeah, these are great speculations, well-informed ones. We just don't have any traction on that, but I think it's a really interesting question. Yeah.
All right, let's thank John for a very exciting forward-looking